Thank you, Mahalaya, for helping us to organize this session. And welcome everyone to our IQ Nucleic Acid Therapeutics Working Groups webinar series, starting with the day one. But before we go into the actual webinar, I wanted to introduce you to the working group and our members. And our mission for this working group has been for the last year and a half to discuss best practices which are related to ADME, DMPK, biodistribution, DDI formulations, and bioanalytical characterizations, as well as the clinical pharmacology strategies of the nucleic acid-based modalities. We did divide these discussions into two different days because of the complexity of the nucleic acid topic. So today on the day one, you're going to hear about siRNAs, antisense oligos, and the related ADME, DMPK, as well as other components of drug development. And tomorrow on day two, you're going to hear more about the lipid nanoparticles, messenger RNAs, in the discussion in a similar webinar. So the objectives of our group has been to understand developability-related challenges for these modalities, and through discussion in our working group, provide clear guidance on the expectations for preclinical and clinical development. And through the webinar today and tomorrow, you're going to see that very intensive and comprehensive discussion around these topics. So we are hoping that we'll all leave with a better understanding on the challenges and some recommendations on best practices. Next slide. So here, I'm just going to show you the number of companies and the subject matter experts that have been involved in these discussions, and how we have brought together all of our thinkings into these complex topics in one place. So I do appreciate that. And next slide just gives you a profile of the panelists from today. I myself, Vibha Java, is the executive director at BMS. Our first speaker is Carol Kuo from Takeda, and she has experience in oligo and cell therapy delivery, followed by Nick Coletti from BMS, who again has 16 plus years developing biologics in different fields, and he has worked on the nucleic acid-based modalities within BMS. Bo Lindmark is from AstraZeneca, and he is a DMPK project lead for these modalities, and has expertise in DMPK drug design, biotransformation, and DDI assessments. Annie Newman, who will be virtually online today, presenting her work from Amgen, and she's also the co-lead for this working group with me. And finally, Amir from GSK is going to talk about the Clin Pharm aspects of the for these nucleic acid-based modalities. So let's go on to the actual webinar, starting with Carol.
Thanks everyone for joining today. Hi, I'm Carol Cole from Takeda. And as Vibha mentioned, many of experts across more than 20 companies are participating in today's working group to summarize today's presentation. And we will be talking about bioconjugation and delivery of oligonucleotides, and how we put together the strategies for bioanalytical, immunogenicity, biotransformation, tissue distribution, computational modeling, and clinical pharmacology. At the end of the presentation, we will open the panel discussions to answer the questions from the audience. And I have been working closely with Rosa Zhang from ASI to put together today's presentation, which is our first topics, bioconjugations and delivery of oligonucleotides. And as mentioned, that oligonucleotide characteristic is mechanism of actions including mRNA degradations, suppression of transcriptions, splicing, and protein binding. the oligonucleotides are typically large hydrophilic polyanion. The size of single-strand antisense oligonucleotides are around 4 to 10 kilodalton. Double-stranded siRNAs are around 14 kilodalton. Modified oligos are not stable. Chemical modifications are mostly on ribose or phosphodiester linker to improve the in vivo stability. Research has been focused on developing a new delivery platforms, including nanocarriers and conjugation to address key questions, key challenges, including large size, anionic charge, membrane permeability, cell-specific intracellular delivery. And in the right figure, as you can see, that we have an examples for the GalNAc conjugates for siRNA and ASO. And for that, we actually, with that, once the conjugates binds, the GalNAc binds to the azo-glycoprotein receptor, which is really abundant on the hepatocytes, will get internalized into the cells and loaded into the endosome. And once the conjugates get into the acyclic compartments, the GalNAc will separate from the oligos, and the siRNA will be loaded in the RISC and bind to the target messenger RNA and further degrade the mRNA. For ASOs, it's going through the similar mechanisms and internal through a receptor-mediated pathway and internalize into the cells and load it into the endosome. And some of them will escape from the endosome and then go into the nucleus and bind to the target messenger RNA, then triggers the RNA-H1 degradation to further degrade the messenger RNA. And it has been demonstrated that oligonucleotide has a transient blood exposure and sustained PD due to the long tissue half-life. This allows an infrequent administrations as quarterly or biannually for siRNA and monthly for ASO. PD time course predicts from animal models as the main driver for first-in-human dose selections. Bioconjugations with a similar chemistries appear to have a similar ADME property, which we are going to give you an examples in the following slides. So here is an example showing you GalNAc siRNA, as I mentioned in the previous slides, through a receptor-mediated signaling pathway are primarily distributed into liver, kidney, and lymph node. And similar study has been done with antisense oligonucleotides. As you can see, the naked ASOs are distributed to many of different cell type. With GalNAc conjugations, it primarily delivers the oligos into the hepatocytes. And this corroborate with the previous slides that shows similar chemistries that demonstrate the similar ADME properties. And delivery technology of oligonucleotides has been developed, has been worked towards several different strategy, including chemical conjugations, which you can conjugate with cell-penetrating peptides or lipids for passive targeting. And covalent conjugate with antibody aptamer specific ligands like GalNAc will be able to actively target specific tissue type or cell type. For this particular chemical conjugations, it's not applicable for plasmid DNA or mRNA delivery, and it requires full chemistry modification. For carrier-based delivery platforms, including LNP peptide complex and polymer-based delivery system, can be able to use with negative charged oligonucleotides and be able to deliver plasmid DNA, mRNA, siRNA, and CRISPR-Cas9. This technology reduced the needs for chemical stabilizations of the charge. With the complexity of these systems poses a new challenge in the development with a perspective to cost, manufacturability, safety, quality assurance, and quality control. And you can find more in these references about the delivery vehicle or delivery technology. And oligonucleotides in the clinical portfolios has been evolved in the past few decades. And it has been, and oligonucleotide has been demonstrated as a promising therapeutics to treat various disease. In the clinic, right now there's 18 oligonucleotide drugs that's been approved and many more is under development. For a portfolio of clinical with oligonucleotide has moving from the rare disease towards a more prevalent conditions. For example, inclisiran is used to treat hypercholesterolemia. And we've been introduced a lot of delivery technology. Most of the approved drug is focused on liver delivery based on the limitation of the technology right now, that we have GalNAc or LNP to deliver the oligos into the liver. As this figure has been shown, like majority of the oligos are targeting to the liver. However, beyond the liver, what we could do is to kind of looking for the new deliveries technology to deliver oligos into CNS systems or muscle eye targetings are under development to really go beyond the liver, deliver oligos to that specific cell type or tissue type. And as an example, this antibody conjugate delivers siRNA to treat DM1 for muscle indications. And of course, there's a lot of early stage development that's in the pipeline, in many companies' pipeline right now. And as mentioned, delivery technology is used to improve the amine property of oligos, and advances in delivery technology allowed oligos therapeutics to go beyond the liver. Multiple consideration needs to be taken into account to adapt delivery technology in oligo drug discovery and development, including altered PK/PD profile, change to tissue distribution, tissue accumulation liability. Of course, when we're talking about antibody peptides or any type of new modality conjugations, non-oligo related safety aspect has to be taken into account. And with that, we do need to initiate new strategies of bioanalysis, biotransformation, immunogenicity, and modeling are needed. With that, I'll hand it to Nicholas Coletti to talk about the bioanalytical strategy.
Thank you, Carol. Let me share my screen. Okay. Hello, everyone. So I'm Nicholas Coletti. I'm a bioanalytical lead working for BMS currently. I am going to be presenting on behalf of all of the IQ members presented here. So I'm honored to do that. This was certainly a collaborative effort, and we all worked on putting these slides together for all of you. Thank you for joining. So as an introduction, we need reliable bioanalytical methods to essentially understand ADME properties, mechanism of action, efficacy, and safety. However, there are various bioanalytical challenges that we face for ASOs and siRNAs. They have high molecular weights, they're negatively and multiply charged, they have high nonspecific binding, they're subject to interference from metabolites, and they may require highly sensitive assays due to their unique PK properties. And not only that, these modalities can also be conjugated. So you can have them conjugated to lipids, sugars, peptides, or antibodies. And furthermore, they can also be encapsulated in LNP particles. So all of this contributes to the complexity of actually measuring your analyte for your studies. So the common bioanalytical platforms that we use to measure these modalities are LC-MS, hybrid ELISA, and stem loop RT-qPCR. So there are orthogonal techniques that we can use as a toolbox for bioanalysis. The choice of the bioanalytical approach should be evaluated on a case-by-case basis. I'll talk about that a little bit later when it comes to the needs of sensitivity, specificity, and measurement of metabolites, the stage of your program, and the regulatory environment. And you would like to keep things consistent across the developmental stages, so you wouldn't really want to change your assay platform throughout studies, such as going through between species and between preclinical and clinical studies. So first, I'll talk a little bit about LC-MS, the assay workflow. I'm not going to dig too deep into the weeds with this, but I will say that this does require a sample extraction step. So that is an additional step that is not required in the future methods I'm going to present. So there are various extractions that can be used: the liquid-liquid extraction, solid phase extraction, or hybridization extraction. We then use liquid chromatography to actually pass the samples through the necessary instruments, and we detect them via LC-MS-MS or LC high-resolution MS. And they all have their strengths and limitations. So the LC-MS platform, it is highly specific, and it is, we've come to a consensus that in this particular team, that it is the gold standard for detecting and quantifying metabolites of these nucleic acid modalities. The method development is very fast. You basically just have to, you really do not have to do any probe design and such. This is just goes right through the instrument, and you're able to measure and get your quantities. It's also well accepted by regulatory agencies, which you'll see in a minute. The limitations, it's relatively less sensitive. There is a lower throughput, and you do need a dedicated machine to analyze your samples. The next method I'll talk about is the hybridization ELISA format. There's various formats that one can use. I'm not going to, again, dig too deep into them, but I will say that the first three here, the one-step, the two-step, and the dual ligation, these methods are typically used for less than a 20-mer sequence. The sandwich ELISA can be used for sequences that are larger than a 20-mer. And I will say that the dual ligation method is the only method here that will measure the intact oligo only. The other three will actually also measure metabolites. And that can be optimized and looked upon and investigated further if necessary. So the strengths of this particular method is it's extremely sensitive. So you can measure in the picomolar, and there's new technologies such as the MPlex technology that can actually measure in the femtomolar range. You do not have to do that sample cleanup or extraction step, and it is high throughput. So you can measure many samples on one particular plate. The limitations is it does have a narrow dynamic range, approximately 1,000-fold. You do have to design specific capture and detection probes. And it, as I mentioned earlier, it doesn't differentiate between attacked and truncated metabolites well. And it's very time consuming to develop the assays. Approximately, it could take weeks to months to develop a validated assay. So the third technique I'll mention is the stem loop RT-qPCR method. This is to measure microRNAs and siRNAs. Again, I won't go too deep into the methods. However, you do need to design a stem loop primer, and you do need reverse and forward primers as well. So ultimately, this method, it is highly sensitive. It's very fast to develop. It's high throughput and highly dynamic. Has a high dynamic range. It is has a low specificity. It is highly variable, and it does require extensive dilutions. I will mention that as a consensus, this is not within the group, this is not typically routinely done, and we do recommend that this method only be used for unregulated non-GLP studies. It has not been used to really present data to regulatory authorities at this point. This slide shows the approved oligonucleotide therapeutics thus far. I'll draw your attention to the platform here, and as you can see, the more recent approvals all have used LC-MS as the preferred methodology, and that is where, as a consensus, we have come to, and this is certainly because one can also measure metabolites if that is a concern. So in summary, the three particular methods are listed here. As I mentioned, the sensitivity is relatively low for LC-MS, high for ELISA, and the highest for stem RT-qPCR. The specificity is highest for LC-MS, low for the next two. Metabolite detection is key, so LC-MS will be the gold standard. You do need to design custom reagents for the hybridization of ELISA as well as the stem loop RT-qPCR. Assay development, we go from fast, slow to medium. Throughput is low for LC-MS. The hybridization is high and medium for stem loop. And as I mentioned, really, the important thing here is that for regulatory, we really have these two platforms that are typically used to present data. And earlier, I did mention that there are ultra-sensitive methods now available. As I mentioned, the NFlex technology, this can measure within the femtomolar range if that is something that your modality requires. So I'm going to change gears now and switch to the immunogenicity strategy. This particular portion of the presentation was completed by Manuela and Swati, so I am acknowledging them here. I am essentially just presenting on their behalf, so I'm honored to do so. So as you may or may not know, these particular modalities can activate the immune system. So they can activate both the innate and the adaptive branch of the immune system. In the innate system, they typically do so through pattern recognition receptors, in particular the toll-like receptors. So these particular receptors are capable of detecting conserved microbial components, such as bacterial cell wall components or viral components. And as we know, viruses can contain double-stranded RNA and genomic materials. In particular, they can detect sequences such as that are rich in cysteine and guanine, and particularly close together within your sequence. So those receptors that recognize this are toll-like receptors 7 and 9. So if these modalities are detected by these receptors, they can activate the interferon pathways as well as the IL-1 beta cascade, and this can lead to immune cell activation. Another component that can be activated is the complement system activation. These nucleic acids can actually bind to factor H, and this factor is responsible for inhibiting the complementary system. Once bound, the complement system can be activated by these modalities. So you would want to assess whether or not your modality activates complement in your toxicology NHP studies. As I mentioned, it can also activate the adaptive system. Here, T cells can be acted directly against peptides or the Fab portion in RNA conjugates. And you can also have B cell responses, your antibodies directed against the RNA or the conjugated moiety that you may have present. So the way that we measure the anti-drug antibodies is typically through ligand binding assays. So here you have your modality is bound to a microtiter plate. Your anti-ASO or nucleic acid antibody will bind to your moiety, and then you detect this with a detection antibody that's conjugated typically to a colorimetric signal that will be generated or an electrochemoluminescent signal. So what is the impact? What have we seen across these particular approved drugs? And what do we see preclinically? Because can we predict what we see preclinically in the clinic? And the answer to that is really variable. So there is limited predictability for ADAs in humans in our preclinical models. Preclinical results have been highly variable in what has been approved. So you really want to utilize a case-by-case approach when it comes to a risk assessment, which I'll talk about later. So in the clinic, what have we seen? So for siRNAs, the incidence is low, so it's less than 5%, and we don't see an impact on PK or PD or safety. So we can categorize siRNAs as a low risk. For ASOs, for different backbones, we do see ADA incidence, approximately 20 to 70% for MOE ASOs, but they are late, so they typically occur 3 months or later. But I do want to point out that there has been little effect on the Cmax, or the area under the curve, and we have not seen an impact on efficacy and safety. And for PMOs, we do not see ADAs observed. What I do want to point out here with this chart, so these are the approved ASOs. I know it might be difficult to see, but I've actually highlighted here that for all of these ASOs, there really has been no clinical impact of ADA on PK or adverse events. So in general, one could say that ASOs and siRNAs would be a low risk for immunogenicity. But obviously, you would want to take your ASO on a case-by-case basis, looking at a risk assessment, which I'll talk about now. So you do want to use a risk-based sampling and analysis strategy when it comes to these modalities. You want to consider the molecule's compound class, how it was manufactured, the length of it, the backbone, any impurities, the dose, the delivery system, and the patient population you're interested in. You want to then consider available data that's comparable to your compound. Preclinically, again, this will be compound and program specific. We suggest that you collect samples for immunogenicity assessment, both pre-dose, midterm, and terminally. And you want to analyze those samples depending on the PK, PD, and safety observations in your study. Again, a case-by-case basis. Now, in the clinic, you want to analyze samples based on a risk assessment. And what I have here on the right is a flow diagram from Nucleic Acid Therapeutics 2022. And here they run down a nice flow of how you should potentially manage your sampling. So from IND to phase 2, they suggest that if your risk category is high, you will collect and analyze a strategy. So you'll collect your samples and you'll analyze independent of any events. If you consider your risk to be low, here you'll implement a collect and hold strategy where you only do the ADA sample analysis if it's triggered by an atypical PK/PD or a safety event. We suggest you collect samples pre-dose and monthly, and then every 3 to 6 months thereafter. And you also want to collect a PK sample at the same time point so you can see whether or not there is an impact. And the optimal timing, depending on the chemical class, on targeted ligands like GalNAc, peptides, and antibodies. So from here, I will hand it off to Bose, and thank you everyone for listening.
Thank you, Nick. I'll share. Can you see my slides?
Not yet.
Not yet. You are sharing, it says. Try again. What about now?
It's in edit mode.
Edit mode. Make it. And now?
Perfect.
Perfect, thank you. So I will discuss biotransformation of ASOs and siRNAs. And I'll do that on behalf of all the 9 members included in this discussion group, representing 9 different companies here. I will start with giving a brief background on common knowledge about catabolism of oligonucleotides. Oligos are catabolized by exon endonuclease enzymes, resulting in chain-shortened metabolites. Catabolism occurs via hydrolysis of phosphodiester or the phosphorothioate in the backbone. Exonucleases remove terminal nucleotides, while endonucleases cleave the oligo in a central position. Nucleases are ubiquitously expressed in blood and tissue. However, there may be different substrate specificity for soluble and membrane-bound nucleases. Their function and substrate specificity are likely to be conserved across species. Metabolism of unmodified oligos is very rapid. It occurs within minutes, and therefore therapeutic oligos are generally stabilized towards metabolism by chemical modification of the backbone. Yes?
Excuse me, but your slides are not moving forward.
Slides are not moving. Oh, I'm sorry. Now? Can you see now?
Yes, thank you.
Sorry for that. They are, I said that they are typically stabilized towards metabolism by chemical modification of the backbone and ribose sugar moiety. Catabolism, commonly the phosphodiester is replaced by a phosphorothioate for backbone stabilization. For siRNAs, common modifications on the ribosyl sugar are introduction of fluorine or methoxy in the 2' position. For ASOs, common modification are methoxyethyl in the 2' position. It can also be constrained ethyl or a locked nucleic acid applied. For ASOs and the GalNAc-conjugated siRNAs, the metabolite burden in liver is much higher compared to plasma. It can be 100-fold or even up to 1,000-fold higher in liver compared to plasma. Metabolites are in general not a safety concern for oligos. At least this working group are not aware of any examples of toxic metabolites of oligonucleotides. The reason for low safety concern with chain-shorter metabolites is that they are expected to have lower affinity via Watson-Crick base pairing to off-target mRNAs compared to the parent compound itself. For siRNAs, active metabolites are commonly seen. On this slide, we provide what is mentioned in regulatory guidance documents regarding biotransformation studies of oligos. The information is very sparse, and there is also some contradicting recommendation in different guideline documents. In the FDA guidance from last year on clinical pharmacology considerations for development of oligotherapeutics, the only thing mentioned with regards to biotransformation studies is that appropriate bioanalytical methods should be used to characterize chain-shorter metabolites. In the FDA draft guidance also from last year on human radiolabeled mass balance studies, it's mentioned that a human mass balance study might not be recommended for oligotherapeutics with known metabolism and elimination pathways based on basic pharmacology and non-clinical ADME information. In the PMDA guidance from 2020 on preclinical safety assessment of oligotherapeutics, it's stated that there is no particular safety concern for naturally occurring nucleic acid components formed by nuclease degradation. They then follow up with saying that, that for metabolites and degradation products containing chemically modified moieties, there is a need to be evaluated for non-clinical safety in accordance with ICH M3R2, similar to conventional chemical products. Finally, in the ICH guidance from 2011 on preclinical safety evaluation on biotechnology-derived pharmaceuticals, it's claimed that the principles in this guidance may also be applicable to oligonucleotide drugs. The message is that the metabolic pathways for this drug modality are generally understood, and therefore classical biotransformation studies as performed for pharmaceuticals are not needed. The Biotransformation Working Group performed a small survey to understand which biotransformation studies are currently performed in the industry. In general, a comprehensive data package is generated on biotransformation of oligonucleotides. The vast majority of us study biotransformation in human plasma and urine, non-rodent plasma, and rodent tissue. Many of us also perform biotransformation studies in vitro, in non-rodent tissue and urine, and also in rodent urine. On this And on the next slide, I will cover a couple of statements or recommendations from the working group where consensus has been reached within all 9 companies involved. The first one is we say that in contrast to small molecules, comprehensive biotransformation studies are not warranted for evaluation of toxicity, in particular for a well-established oligonucleotide platform. For ASOs and GalNAc-conjugated siRNAs, the metabolite burden is in general much higher in liver than in plasma, and therefore liver is the most relevant matrix to study biotransformation. However, there is in general no access to human liver in vivo. In humans, we have access to plasma and urine. The metabolite profile in plasma mainly reflects the parent oligo and metabolites formed by elimination of one or a few nucleotides from the terminal end. In urine, there is a bias towards smaller oligofragments, and therefore utilizing both plasma and urine for metabolite studies will provide a more complete picture of the oligonucleotide metabolic fate. For biotransformation studies in animal tissue, it is from a 3R perspective recommended to utilize terminal samples from toxicology studies as far as possible. Also scientifically, it can be argued that metabolite profiling studies after repeated dosing is preferred rather than after a single dose because of the slow redistribution and elimination of an oligonucleotide. In vivo, last bullet on this slide, in vivo, in the absence of human biopsy, plasma metabolite profiling and characterization may be of value for confirmation of previously identified metabolites and to confirm cross-species biotransformation pathways. In vitro studies may be of value. Systems which are commonly used are, for example, liver S9, liver homogenate or hepatocytes for cross-species comparison and for early identification of potential active metabolites in human. If using in vitro studies, there is a need to build platform-based knowledge and confirm in vitro to in vivo correlation. From a 3R perspective, the thrive should be to replace in vivo studies with in vitro as far as possible. There is in general a limited value in biotransformation studies using radiolabeled compound. In contrast to small molecules, the potential metabolites formed via backbone hydrolysis are predictable. Much the same information can be obtained without a radiolabel using LCMS and access to synthesized metabolite standards for quantitative assessment. For novel chemically modified nucleosides, conjugates or linkers, for example, lipid conjugates or novel linkers of GalNAc, the metabolic fate of that molecule needs to be understood. A final slide from this working group is covering arguments for why there is, in our opinion, a limited value in conducting MIST for oligonucleotides. MIST stands for metabolites in safety testing. It is stated in the FDA MIST guidance that it applies to small molecule non-biological drug products. And I start with here, have listed a few comments from the MIST guidance as a recap what it is all about. First, it covers recommendations to the industry on when and how to identify and characterize drug metabolites whose non-clinical toxicity needs to be evaluated. Drug plasma concentration and systemic exposure in the non-clinical studies are compared with systemic exposure in humans. Exposure should be at steady state unless there is some justification for different measure of exposure. I have a couple of reasons here why our view is that MIST evaluation of oligotherapies is of limited value. Oligos are metabolized by nucleases, which has much higher cross-species similarity compared to conventional drug metabolizing enzymes. Nuclease-mediated metabolism result in chain-shortened metabolites, which are expected to be less toxic than parent compound. The propensity to hybridize via Watson-Crick interactions decrease with decreasing chain length. And as I mentioned before, this IQ working group is not aware of any examples of safety risk caused by metabolites of oligonucleotides. In contrast to small molecules, the metabolite burden of ASOs and siRNAs, if they are gamma conjugated, is much higher in liver than in plasma. Furthermore, metabolite profiles in tissue is not necessarily reflected in plasma. That was all on biotransformation, and I will now continue to discuss tissue distribution on behalf of the 7 members listed on this slide. So we have here done, similar to the biotransformation group previously shown, we have extracted what is mentioned with regards to biodistribution studies in regulatory guidance documentation. In the FDA 2022 draft guidance on clinical pharmacology for oligotherapeutics, it stated that tissue distribution is determined by the chemistry of the oligo, both the backbone and conjugation, drug target, plasma protein binding, and route of administration. These aspects therefore need to be taken into consideration. Nothing related to tissue distribution is mentioned in the PMDA 2020 guideline for preclinical safety assessment of oligos. In the ICH 2011 guidance on preclinical safety evaluation of biotechnology-derived pharmaceuticals, which may also be applicable for oligodrugs, it is stated that tissue distribution studies in relevant species are useful. They also comment that if using radiolabeled compound to study tissue distribution, data may be difficult to interpret due to rapid in vivo metabolism or unstable radiolabeled linkage. Biodistribution studies can be performed by utilizing non-radiolabeled or radiolabeled compound using quantitative whole body autoradiography detection. For studies with non-radiolabeled compound, tissues to be considered for analysis includes high exposure and high target expression tissues. A qualified LC-MS method or an LBA method can be used for quantification of unconjugated ASO or antisense strand in selected tissue homogenates. From a 3R perspective, tissue samples in tox studies of determination may be used. By including a separate group of animals, tissue exposure can be measured for a given recovery period after the last dose, and that is to understand any risk of accumulation of any form metabolites in tissue. Using radiolabeled compound and QWBA detection is a well-established methodology to study biodistribution of small molecules. It provides information on distribution profile of all drug-related material over time in the whole body. To avoid biased results in the QWBA study, the radiolabel must retain the specific radioactivity in the parent compound, which can be a challenge for radiolabeling of oligonucleotides. Different radiolabeling strategies have been used for oligos. For example, uniform labeling with sulfur 35 in the phosphorothioate backbone, single or dual labeling with tritium in 2' or 5' position of the ribosyl, hydrogen and tritium exchange of the purine base, or carbon 14 labeling of a thymine base. These strategies are associated with the risk of biased results mentioned before, in case the specific activity of the metabolite formed is altered, which could be a potential risk in case of uniform labeling, or if metabolism results in cleaved product not retaining the radiolabel. which is a potential risk with single and dual labeling, or exchange of tritium to water needs to be avoided, which is a potential risk when using tritiated purine. The final slide on tissue distribution covers a proposed decision tree from this working group on the extent of tissue distribution studies that are needed depending on chemistry used in the oligonucleotide. Before going into this decision tree, just a few comments and thoughts about need of QWBA studies for oligos. A QWBA study is an essential enabling study for a conventional ADME trial to support human radiation dosimetry. However, it is a gold standard for small molecules, or hence it's a gold standard. From a regulatory perspective, ASOs and siRNAs fall under the category small molecules. However, as human ADME trials are generally not expected for ASOs or siRNAs, and I now refer to what I mentioned before, the FDA 2022 guidance on human ADME studies, there is no regulatory requirements that biodistribution studies must be conducted with radio-labeled material. Alternative non-radio-labeled approaches can be justified. There are pros and cons of conducting biotransformation studies with radio-labeled ASOs and siRNAs. Looking in this decision tree, what we propose is that in case the oligo does not contain novel chemical modifications, ligands, or formulations, a cold study with limited number of tissues of interest, could be target tissue, high exposure tissues, may be warranted. On the other hand, if the oligo contain novel chemical modifications, then a cold study with multiple tissues or a radiolabel study may be warranted. And that was all from me, and I will now hand over to Annie, who will discuss computational modeling. Thank you.
Hello, everyone. This is Annie Luman from Immunogen, and our next topic is going to be computational modeling and quantitative approaches as it applies to siRNAs and ASOs. And the thoughts summarized here are that of many experts that you see listed here across 10 participating companies. And so we wanted to start off by emphasizing the need for computational modeling and simulation approaches for these modalities. As you might already know, the tissue PK, but not the plasma PK, is a primary driver for PD. And you can see an example of that for GalNAc siRNA in the figure to the left, where the plasma PK is shown in blue and the PD time course is shown in purple. And you can see the temporal disconnect between plasma PK and the PD time course. On the other hand, the total tissue concentration that you see here in red and green are in better alignment with the PD time course. So the goal of these modeling approaches is to bridge this disconnect that you see between plasma PK and PD by predicting the target tissue concentrations. But because we see that there is a lack of tissue PK in humans, these type of modeling approaches also become useful in leveraging the available tissue PK data in non-clinical species for translation, which can be then used to guide FIH study design and dose selections, and also for various clinical applications, including SAD/MAD dose projections, and for evaluating patient factors, as we would discuss more in detail shortly. So there are several considerations that need to be kept in mind for the development of such models. First, specifically to capture the modality-specific mechanisms that focus on elaborating the tissue distribution. But in addition to which, The key considerations involve asking ourselves questions such as, can we leverage any legacy data that's available? Is the target in the siRNA sequence active in the test species? What would be the biomarkers that we can measure? How would we tie a serum biomarker to a PD endpoint? And addressing these considerations and having in mind to build the framework would be helpful in developing a successful PK/PD model for these modalities, and then leveraging the data in the various animal models and making reliable predictions in humans. The complexity of these tools can vary from, say, non-clinical, non-compartmental analysis to compartment modeling, physiological models, and quantitative systems pharmacology models. And the data availability and the question that we are using this model to address would drive the choice of the model complexity. For example, whether are we just trying to make steady state predictions, or are we trying to predict time course and estimate dosing scenarios? Are we trying to do translation or predict disease states and life stages, or test disease and treatment hypothesis? So it depends on the question that you're asking to decide on what level of complexity you want the model to be. But what is currently done is what we have summarized here. If you look at the table to the right, it shows the type of models that have been submitted in regulatory bodies for approved siRNAs, and they primarily are population PK/PD models, dose or exposure response models. And in practice, a current drug development practice, both population PK modeling and PBPK type modeling approaches are successfully applied for both translation and clinical applications. In the case of translation, it's used for allometric scaling, FIH dose predictions, both from a safety approach and a translational approach. And then also for clinical applications, it spans for SAD, MAD dose projections for estimating the starting dose and the final efficacious doses, and designing these studies in both healthy volunteers and patients. Followed by model applications that also include dose and dose regimen optimization, and other models such as concentration QTC modeling could potentially also support waivers. And furthermore, we could also use these models to inform dosing considerations in organ impaired populations in lieu of conducting a dedicated study and potentially supporting a waiver as well. So we wanted to discuss a little bit more detail about the capabilities and considerations for pop PK/PD and semi-mechanistic PK/PD models for siRNAs and ASOs. So from what we have discussed, we've noticed that the model structure for ASOs and siRNAs and GalNAc conjugated ones are pretty much very similar. They're primarily compartmental in nature, with the goal of predicting the concentrations in tissue and peripheral sites, which are the site of actions that drive the PD. And if needed, additional transit or effect compartments to capture any delayed transcription or degradation of ASOs or siRNAs can be added. And for the PD component, indirect response model seems to capture well the delayed PD response due to downstream delay in translation and PD turnover. Some exceptions include local delivery. If you're having intrathecal administrations, then in that case, we don't need specific compartments to describe the redistribution between the tissues. In general, what we noticed was that there's good translation of PK from non-human primates to human, having in mind that the PD translation may be target dependent. So far, most of the modeling approaches have been in the compartmental PD modeling space, but PBPK modeling approaches can become important when multiple target organs become involved, enabling projection of PK/PD in these target organs where data might not be available or accessible. And it also allows for capturing physiological species differences to support translation and evaluate patient factors as well in the case of pediatrics and organ impairment for extrapolation type applications. Here, for just references and knowledge, we have shared two examples of PBPK models. One is the intrathecally administered ASOs, and another one to the right is a subcutaneously administered GalNAc siRNAs, for GalNAc siRNAs that are currently available for application in the field. So knowledges and data gaps do exist. This is still an evolving field. Particularly, there's additional research we felt is needed around the mechanisms and kinetics of tissue uptake, distribution and clearances, and risk loading kinetics, specifically for siRNAs. There's also a lack of physiological information on key mechanistic components that are relevant to ASOs and siRNA, for example, nuclease and ASGPR expression, target turnover rates, ontogeny, disease, sex, and species differences. There is still room for more information. There is also limited tissue PK data in preclinical models that could pose additional challenge for model construction. For example, in our discussions that came about that terminal half-life, if it's not well captured, it could be a challenge to translate. And also, some of the liver targets, if it lacks circulating biomarkers, it adds an additional layer of complexity where it warrants now liver biopsies and animal models to be able to measure tissue mRNA and target protein reductions. Moreover, because tissue data in the clinic is not normally accessible, this poses some challenges on model verifications as well. So not all models are perfect, but some are indeed useful as it still holds in the case of these modalities as well. So in summary, the best practices and recommendations are that the computational modeling approaches have indeed been successfully applied for translation and in the clinical context for both siRNAs and ASOs across indications. The model structure and the mechanistic complexity depends on the data availability and what we are wanting to use the model for. Current strategies are primarily in the fit for purpose mode, but it is assuring to know that compartmental models and first order kinetics and indirect response model in general are useful, are successful to describe the PK/PD relationships. This is more of a general context, but when you go into conjugated oligos, there might be exceptions where additional insights for model constructions are needed, for example, incorporations of more mechanistic information for target tissue uptake, or incorporation of some nonlinear kinetics could be needed. And finally, more mechanistic approaches such as PBPK are available, and such applications for the purposes intended would become, as the complexity of the modeling need increases and more additional mechanistic data emerge, such applications will become more prevalent, going beyond the fit for purpose context. And the data limitations, as we discussed, do exist, and the knowledge gaps that we have identified could guide effectively further research. With that, I'll stop this section here. Thank you.
Amir, we can't hear you.
Apologies. Hello, everyone. This is Amir Youssef from Clinical Pharmacology Modeling and Simulation at GSK. I will discuss the clinical pharmacology considerations for oligonucleotide therapeutics. This was a collaborative effort with many SMEs in the field as listed in the slide here, and I'm very happy to present on behalf of this sub-team. The clinical pharmacology consideration in this will cover three topics: the characterization of QTC prolongation, the evaluation of drug-drug interactions, and the assessment of hepatic and renal impairment. These three topics, in addition to the immunogenicity, which was discussed earlier in this webinar, were covered in the FDA draft guidance that was issued last year. The draft guidance on oligonucleotide therapeutics cover both antisense oligonucleotide and siRNA. So starting with the first topic, which covers the QT prolongation, the table here actually lists in the upper half, you can see the antisense oligonucleotide, and in the lower half, we have the siRNAs that are currently approved from the FDA. This is the majority of the drugs that were approved. It's not a comprehensive list since some were approved very recently. However, all this information was collected from the FDA review file, the prescribing information, and the scientific publication. As you see here, there is, of course, regardless of the modification or bioconjugation that is done for the ASOs or siRNA, regardless of the route of administration, we can see here that the hERG assay was approximately performed for about half of these cases. We have seen that every drug here has some sort of non-clinical ECG monitoring was performed. This can be either a toxicology or safety pharmacology studies. Clinical ECGs also, those were performed across all cases here. These are concentration QTC that was performed. Only in very rare cases, there were actually a dedicated TQT study. The overall conclusion from this study is that there is no apparent effect of QTC interval prolongation was observed, and none of the labels actually describe a clinically relevant prolongation of QT interval. So moving back to the guidance, as we mentioned, there is no effect of oligonucleotide therapeutics on QT interval in the small number of drugs that has been done. However, according to the guidance, the current clinical experience does not adequately support an overall conclusion. This is the language in the guidance. And then the FDA actually recommend two things: the assessment of QT prolongation list and a proposed QT assessment plan that should be submitted and discussed with the agency. To streamline this process, the IQ clinical pharmacology subgroup has some recommendation. Actually, they comprise from two parts. The first one is to conduct non-clinical ECG evaluation. These are high quality ECG in toxicology study and possibly hERG assays. To complement the non-clinical ECG evaluation, clinical ECG evaluation also is needed. The best time is to conduct these or collect this time PK time match high quality QTC data in triplicate during the SAD and MAD. And this is because this study actually provides a widest range of dose and provide a wide range of concentration. So it's actually a benefit to conduct this assessment during the early in SAD and MAD and use do the PK/PD data analysis and use this as a basis for TQT waiver. This is the recommendation. To some, the oligonucleotide have a very low potential for QT prolongation. There is no relevant QT prolongation that has been observed for all approved oligonucleotide up to date. It's of course being treated as a small molecule in regard for QT evaluation, and then of course maybe down the road there will be different, as the knowledge build up, might be some revision on how these study might be some relaxation of the language a bit. So that covers the first topic in clinical pharmacology. I'm moving to the second topic, which discusses the drug interaction. As mentioned earlier, all these oligonucleotide therapeutics are not typically metabolized by CYP enzymes. They're actually being metabolized mainly by endo and exonucleases. And therefore, we do not expect or anticipate they will be any inducer or affected by any inducer or inhibitor of CYP enzymes. Also, they are not affected or impacted by transporter modulation. As you see here, the top table shows the ASOs, and the bottom table shows the siRNAs. Regardless of all of these, there was maybe most of these has conducted an in vitro CYP studies, which again probably is very common from the small molecule world. Very few has a dedicated clinical trial. Mipomersen B1 and Givosiran is another one that has actually conducted a dedicated DDI assessment. As you see here, there is no actually any impact in the label that any of these has any significant DDI. So regardless of in vitro or even in vivo, there is no indication of any significant DDI. There was one DDI that was found to be significant, and that happened with givosiran. And this actually, it was a PD mediated, because this drug actually is a drug that through pharmacology leads to a decrease in the heme synthesis and decrease in the CYP enzyme level and activities. So that was actually not a drug itself. It's mediated by the biosynthesis pathway, and even this, in this case, the in vitro DDI did not reveal any drug-drug interaction. So it was a PD mediated. It has to be done through the strong understanding of the pathways and the biosynthesis pathways. So giving all the accumulated information and the class property, our recommendation here is to actually, the in vitro study are not warranted, and they can be determined by case-on-case basis. However, at this point, and in the light of the draft guidance, an alignment with the regulatory agent is needed. This might again change down the road as more and more information is built, but we think that we have enough information now that shows that in vitro DDI are not warranted, but again, an alignment need to be achieved with the regulatory agent. Another thing is of course, the human DDI has been conducted, has not revealed anything, except with this case, which was again not a PK mediated, it was a PD mediated. We need to, for every drug, a mechanistic-based effect as a drug disease interaction, all of these need to be well studied to understand if there is any potential for something like in the givosiran case where a PD mediated interaction can occur. Moving to the last topic here in the clinical pharmacology discussion, which is on the renal and hepatic impairment. So it's very important to understand the impact of hepatic and renal function on the PK, PD, and safety of oligonucleotide therapeutics. With safety in mind, it is best to enroll different degree of impairment, either renal or hepatic, and to utilize the modeling-based or a POP-PK approach to try to understand the impact of impairment, either renal or hepatic, on the exposure. So as you see here, the majority of the drugs that has been approved, either ASOs or siRNA, has utilized a population PK model where they kind of use a different degree of impairment as covariate in subsequent analysis. There is very, very few cases where actually a phase 1 dedicated renal or hepatic study was conducted. The overall finding in this is that there is no apparent clinical relevant effect of mild or moderate organ impairment on oligonucleotides. So what is our recommendation for hepatic impairment? It's very important to understand the role of liver and kidney in the disposition and elimination of the drug. When oligonucleotides are not predominantly renally cleared, which is like has a threshold about less than 30%, or is not targeting the liver, it's best to enroll subject with a full range of renal and hepatic function in the late clinical phase trials and use modeling approaches. When oligonucleotide is substantially renally cleared, of course, there is a further characterization need to understand the impact of renal impairment. If it's actually targeting the liver, we recommend to use sequential enrollment. So you start with a mild, then we can do the moderate to understand all of the impact of the impairment on the exposure and PK/PD of the drug. Of course, the pharmacodynamic assessment with POP-PK, those are best way to analyze the impact, as we mentioned before. The last resort is to resort to a dedicated single dose phase 1 renal impairment or hepatic impairment studies. If that is the case, if that's kind of last, like the sponsor need to conduct this, we recommend a reduced study design that is acceptable if, again, the modeling approach and enrolling subject in the late phase trial was not feasible. that concludes the clinical pharmacology section. And now I would like to actually go to the key messages of different sub-teams. So starting from the bioconjugation, multiple considerations need to be taken along with drug discovery and development for oligo when adopt conjugation technology. Those include altered PK/PD profile, tissue accumulation liability, non-oligo related safety aspect, etc. For bioanalytical, the choice to use LC-MS or LBA PK methodology for oligo should be considered on a case-to-case basis. The majority of FDA-approved oligonucleotide utilize actually the LC-MS as a preferred method for quantitation and metabolite detection. The LBA ultra-sensitive method has been recently developed to detect oligonucleotide in extremely low quantities if such measure is required. For oligonucleotides, for immunogenicity, the immunogenicity risk evaluation should be performed, which considers the molecule attributes. The human predictability using preclinical assessment of approved oligonucleotide was variable, and clinical EDA sample analysis should follow the collect and analyze or collect and hold paradigm based on the risk associated with the molecule. For biotransformation, the liver is the most relevant matrix to study biotransformation for ASO and GalNAc siRNA. The missed assessment of this platform was of a limited value. For tissue distribution, for both ASOs and GalNAc siRNA, a cold study investigating limited tissue, more or less or equal than 2, may be warranted. For a non-platform, a cold or radiolabel study investigating multiple tissue more than 2 may be warranted. For computational modeling, PK/PD modeling tools recommended to be leveraged for translational, like first time in human and dose projection, and for clinical application like dose and dose regimen optimization, and for organ impairment. Model can be either fit for purpose or can be more mechanistic depending on the application need and the data availability. Lastly, for Clin Pharm, based on the historic data and class property, ASOs and siRNA are unlikely to show QTC prolongation. However, at this time, a QT study waiver can be based on non-clinical and clinical, like QTC, consultation QTC evaluation. There is a very low potential for drug-drug interaction, and we need to lever modeling approach in an adaptive phase 2 and 3 to assess the effect of renal and hepatic impairment that need to be done. With this, I will turn it back to Veba to start the panel discussion. Thank you.
Thank you, Amir. And I would first of all like to thank all the speakers and also the team who got all these slides together today for us to see a very comprehensive look at the antisense oligos and siRNA from our ADME and Clin Pharm perspective. So before I start, I saw some questions in our chat, and I would encourage our audience to write their questions in the chat so I can use them to direct to the panelists here. In the meantime, I did see a couple of questions. First of them, I think Mahalea did try to respond about sharing of the slides. And so we are planning to convert this webinar content into 2 white papers, and those are, the team is working on it as we speak. So you are going to see that content in these white papers in the next, hopefully soon, the next few months. I'm being very ambitious, but also the recorded slides are available for people to audit. So please stay put on like looking at our white paper, which will be more comprehensive than these slides. Now, I can start some of the, I saw one question and I feel Amir did answer it in his last 2 slides, but it's about it's about the ASO metabolism and impact of hepatic impairment. So Amir, if you have any more thoughts on it, or even Bo, if you have any thoughts from a biodistribution perspective, please go ahead and maybe respond to this question.
Yes, Veba. So of course, the modeling approach would be the preferred one to understand the effect of hepatic impairment. That's of course in we see like in many GalNAc where there is a hepatic impairment, less tendency for the hepatocyte to uptake the molecules, and then we see an increase in systemic exposure. So that is better addressed. Of course, there is a room for a dedicated phase 1, but most of these cases can be addressed by enrolling a different degree of impairment and be able to assess that during the modeling using a POP-PK as a covariate.
Thank you. Bo, did you have any thoughts on it?
If the question was about any change of nuclease-mediated metabolism in hepatic impaired patients, we have not studied that in AstraZeneca. We haven't done any metabolism studies in hepatic impaired patients. But I wonder, because what I mentioned also in my talk is that the most value of studying metabolism biotransformation is liver, not plasma. Typically in plasma, we see very few metabolites formed. And thinking of potentially looking in preclinical species in tissue and any change in metabolite profile, that is something that I guess could be considered, and maybe somebody has experience of that. I don't, unfortunately.
There's another question around biodistribution, and maybe you can stay online, Bo. So it's siRNA has short half-life in blood, but long residence time in tissues. Where is the siRNA sequestered in the tissues to protect against degradation? And Carol, I see you responded. So both of you can take a stab at it.
So it's basically based on the case of GalNAc conjugations, which allows oligonucleotides to deliver into hepatocytes in the liver very quickly. So that's why we do oftentimes seeing transient exposures. And yes, for the long half-life, it's actually due to first chemical modification is kind of stabilized the oligo. And of course, slow release of oligonucleotides from the endosome is one of the key contributions to the long PD half-life.
Bo, did you want to add something to this?
No, nothing to add. I think that was clear. Thank you, Carol.
Great. Maybe I'll ask one question, and that came up, Carol, when you were talking about where do we see the nucleic acids go to, and you mentioned lymph nodes, and that was very interesting for me, because for me, lymph node is a place where you also educate or modulate your immune system, right? And we talk about immunogenicity. So between you and maybe Nick, could you talk about what that impact would be if we are expecting these nucleic acids to reach the lymph nodes?
I guess with that, Nick was kind of giving a very comprehensive talk about the immunogenicity for oligonucleotides. With that, from my perspective, I think for most of the conjugations need to be taken into account. If there's any exposures in the lymph nodes, then that might potentially trigger some immune response in that way. And that's kind of based on some of the antibody case or some of the specific modality conjugations that could potentially trigger the immune response. And for the oligos, that Nick could kind of elaborate a little bit more about the immunogenicity over there.
Yeah, sure. So yeah, if they are indeed getting into the lymph nodes, I think your biggest idea here would be B cells, because there's a lot of antigen presentation there. So toll-like receptors, right? So I think really the take home here is to prevent activation within the lymph nodes, you would want to design your oligo appropriately. So to steer clear of TLR9, if you can design your oligo to not have cysteine or guanine at all, that's something I've dealt with in the past personally. That is kind of your best bet to prevent activation of TLR9. Also, again, to also prevent activation, you'd want to restrict the size of your oligo. So it's been shown that the larger oligos can be more immunogenic. So you want to keep the size of the oligo as small as possible. So yeah.
Thank you, Nick. There's one more question I'm hearing that some people are having trouble putting their questions in Q&A, Mahalia, so I don't know why, but this question is about data. What data should be submitted to regulators for a waiver of the human mass balance studies, especially if QWPA is not conducted? So I will let one of the panelists, I keep on asking Bo, but I think it's your question, Bo, or maybe Carol can answer.
So I don't think you need so much argumentation actually, because we have experience across this working group not generating QWBA data for ASOs and siRNAs, and there has not been challenge back to our experience. So in my experience, that is not warranted by the regulators to see that data.
So the human mass balance studies waiver, we don't need to ask for that. I mean, especially if the QWP.
So in my experience, we in any pre-IND meeting or any opportunity we have with interactions with the regulators, we provide our standpoint to waive these studies, and we have not got any challenge back on that so far.
All right. So another question, Carol, I know you were responding to it in the chat. I'm just going to speak it up is, what does exposure to a specific tissue always mean that you get knockdown of your messenger RNA target?
Sorry, Viva, can you say that again?
Sure. Does exposure to a specific tissue always means that you get a knockdown of your messenger RNA target?
So it really depends on the modalities and also the potentiation of the oligo. And of course, cell-specific targeting strategy is usually kind of like seeing, sometimes we do see productive uptakes or non-productive uptake, which will give you an exposures in the specific tissue type. However, those cell types are either not expressed that high of the level of your target genes that you might potentially see a high exposure, but no knockdowns. So it really depends on what type of targets that you are looking into, and whether it's going into like through the active productive uptakes or non-productive uptakes. So for productive uptake, of course, you will see the correlations between the exposure versus the knockdown efficacy that you will see in the specific tissue type. But for some of the tissue type, it will kind of like taking up the oligos, but it's not potentially in the way that we will see the knockdown.
Thank you, Carol. And two more questions, one about toxicity concerns on GalNAc accumulation in the liver. Who can talk about that? Anyone from the panelists who want to speak to the toxicity concerns on GalNAc accumulation in the liver? Bo, do you want to speak to that?
Is the question related to the GalNAc is a natural compound, no big concern with GalNAc, or is the question more maybe related to the linker associated with it, which could be a different thing? I don't think GalNAc per se is a concern.
Okay, thank you. And the second one is there a gold standard for quantification of GalNAc linked siRNA and its metabolite? Nick, do you think you can respond to that?
Wen Yang, can you respond to that?
Yes,
Sorry.
sure.
Thank you.
Yeah, so for GalNAc conjugated siRNA, typically we use LC-MS. So siRNA, the active species is the antisense strand, while the GalNAc is conjugated to the sense strand. Typically for quantitation, we measure the antisense as active moiety, while for the GalNAc conjugate sense strand, we can measure it. Usually, once it's in the liver, the GalNAc fell off very rapidly and it got metabolismed. And you can use LC-MS to detect its metabolite for metabolite profiling.
Thank you, Wenjing.
Sure.
One more question. We are getting a lot now, and we are almost ready to close here. Are there any useful in vitro methods to assess uptake and release rates from endosomes? I'm just opening it to whichever panelist wants to take this. If we cannot, we will address it and then maybe respond to it separately, because there are questions that we may want to take back to our team. We can do that. For oligos with novel bioconjugation, human ADME study might be needed. I don't know if that's a comment or more like a question. So
I think that is a valid question, and that is something we need to work out. One thing is, when we work now, most of us have a lot of experience with GalNAc conjugated siRNAs and ASOs with well-defined chemistry as well. That is a different thing. But when we start to move on to novel chemistry, novel conjugates, linkers, then I think these aspects need to be considered carefully.
all right, so we are almost at the end of this session. These are very good questions, and please reach out to us, to me, if you want, and we can answer questions which we may not have covered right now. And I'll give it back to Mahalea because we are at 1 minute to the end of this session.
Okay, great. Thanks, Vibha. Thanks to all the speakers and panelists who participated today to present this very, very important and interesting material. And mostly thank you to all of the audience who joined us for today's webinar. If you're still with us and you have not yet registered to attend tomorrow's webinar, I encourage you to do so. You will be able to do that via the same link that you used to register for this one. And so until then, we'll see you tomorrow morning at the same time, and we look forward to sharing with you more interesting material on nucleic acid therapeutics. Thank you very much. Take care all. We'll see you tomorrow.